Welcome back. We are going to start with the chapter 33 and we are going to discuss about the adaptive immunity. We are only going to talk about the chapter section number 33.1 to 33.8. The 33.9, we will just only skip that part. The first thing that we are going to talk about is the relationship of the adaptive immunity. One of the very important concepts of the adaptive immunity that relies on the recognition as well as the memory. And we are going to compare and contrast it with innate immunity that we have learned in the last chapter, that how it differs or how it complements the innate immunity. One of the very important part that is very important, once our innate immune system is breached, at that time, the adaptive immunity takes over. So usually most of the times that if there is any kind of pathogen that get past your first line of defense, which are the physical and the chemical barrier, then those are attacked by the second line of our immune defense, which is also the part of the innate immunity. And those are your macrophages and the neutrophils or the natural killer cells. We have talked about that. Somehow, if the pathogen even breaches those, that means that the pathogen could not be controlled by the second line of defense, which is the last line of defense of the innate immunity. At that time, the adaptive immunity is needed to be triggered and ready to go. Most of the times, when there is a pathogen invasion, already the innate immunity will inform the adaptive immunity that there is something going on. But the adaptive immunity takes about 5, 7, or 10 days to kick in. But once they understand that the innate immune response could not check the pathogens, they will immediately spring into action and work. And we are going to talk about that in this chapter. So let's take a look at the adaptive immunity. This has three major functions. It recognizes non-self. What we mean by the non-self, that if there is a foreign antigen coming in, it should be recognizing that non-self. And we're going to talk about that in detail. Respond to non-self, what we mean by that, the effector response means that it will eliminate or renders foreign material harmless. This could be a pathogen, that could be a molecule, which could be a toxin, it could be anything. We are going to elaborate that in detail. And along with that, what is going to be important, and that was not the part of your innate immune response, but this is a sole property of your adaptive, or we call this acquired immunity, which is the memory cells remember the foreign material, and upon a second encounter with the same pathogen, the immune system mounts a faster and more intense response. And that is the reason that most of the time the vaccines work. So while the vaccine goes into your body, this is considered as the foreign material. You develop an immune reaction. And when your real pathogen affects you or just tries to infect you, most of the time your immune system will be able to counterattack that and you will not have the infection due to that memory. And also very important part is that remember non-self. This is the same part of that. So these are the major functions of your adaptive immunity. Then there are some characteristics of the adaptive immunity. The most important part is the discrimination between the self and the non-self. Usually responds selectively to non-self, producing specific response against the stimulus. Why it is important? Because remember that we have thousands of these different proteins in our cell, and by chance, if the regulators, particularly the T cells or the B cells of the adaptive immunity, by chance react with them, that will be detrimental to our own body because these proteins are present on our cells and it's going to attack them. So all the regulators or the cells which help in understanding the non-self make sure that there is, during the development, no T cells or the B cells develop which react with any of the self antigen. And we have already talked about that. We're going to talk about that a little bit more detail, that during the development, if any of the immune cells of the adaptive immunity react with any of the self antigen, means these are the proteins of your own body, they are clonally deleted or they are killed. The second important part is the specificity. This can be directed against one specific pathogen or foreign substance. And this number is mind-boggling, that the antibodies of different specificities which can bind to this, or the B cell receptor that can bind to these different types of specific things. We're going to talk about and elaborate about those things. These combinations are trillions. What do we mean by that? That your body can respond to trillions of different kinds of three-dimensional molecular shapes come in all varieties, which can be different molecules, toxins, pathogen, or pathogen-related structural surface. So basically, your adaptive immune cells do have receptors in the combination of trillions to understand all these different three-dimensional antigenic structures, which we called as an epitope. So that is a very amazing thing of the adaptive immunity. The diversity is also generates enormous diversity of cellular receptors and the antibodies. So again, the specificity is also very, very high, and the diversity, enormous diversity of cellular receptors and the antibodies. And we are going to talk about that how many different types of cellular receptors. We are going to talk about the T cell receptors, that we're going to talk about the B cell receptors. Then if you just think about the antibodies, there are many different types of antibodies, each have different functions. So, and they are very diverse as well. We are going to explain that in detail. And also that it responds to a second exposure to a pathogen is fast, so that there is no noticeable illness that we already talked about. So now the types of adaptive immunity is the humoral versus cellular. What we mean by that adaptive immunity has two parts. One is a humoral immunity, and also called the antibody-mediated immunity based on the antibody activity. And humor means fluid. The reason is that historically, all these different antibodies were isolated from the blood serum, and these antibodies are present in your blood serum, which is the liquid part of your blood. If you take the blood cells out, the serum that you have, the antibodies are present in that serum. And the serum is a fluid that is humor, and that gives you the humoral immunity. The second type, other than the antibodies, which are secreted by specific cells of your adaptive immunity, is the cellular immunity. The cellular immunity is given by another cell, which is the T cell. And this is also known as a cell-mediated immunity based on action of specific type of T lymphocyte. And we are going to see that there are several different varieties of the T lymphocyte. There are some T lymphocytes which are the helper ones, which activate the immune system. Some regulates the immune system. And then there is a special type of T cells, which we called as a cytotoxic T cell. We will come back to that over and over, but we call them as a cytotoxic T cell. And the function of those specific cytotoxic T cells is to provide the cellular immunity, means it is a direct killing of the cells of our own body infected by a pathogen. Why this is important? It is important because there are sometimes that is the only way. Because sometimes if there is a virus infected cell, the virus is replicated inside the cell, there is no way that you can go inside a cell and specifically kill the virus. There is no mechanism to do that. So the only mechanism is that a cytotoxic T cell will kill the whole cell infected with the virus or the specific pathogen that it has. And along with that, it will be able to kill that pathogen as well. So on the other hand, you can just think about a small thing that once the cell is dead, lysed, there could be virus particles still inside the cell that may come out into the body fluid or may come out in the blood or in the serum. At that time, remember that your humoral immunity is present in your body fluid, on the mucous membrane, in the blood, in the serum. So all these pathogens which are coming out of that dead cells after lysis can be taken care of by this antibody-mediated immunity. So each one of them complement each other. So next we are going to talk about that the antigens elicit immunity, and they predict the types of molecules that can serve as an antigen. And we are also going to talk about some of the concept of what is hapten and the relationship with the haptens to the antigen. So the antigens are non-self substances that elicit an immune response or an immunogen. It must be a large complex molecules, and each of these antigens do have some determinant sites which are called epitopes. And the epitope, this word is very important, is a site on the antigen that react with a specific antibody. What we mean by that, suppose there is a particular protein which is antigenic. A single antigen, again, make sure that you understand that, a single antigen may have multiple epitopes. And usually if that has multiple epitopes, we call that as an multivalent antigen. Whereas on the other hand, if there is an antigen which does have just only one epitope, only one site on the antigen reacts with a specific antibody, we call it as a monovalent antigen. So of course, always the multivalent antigen, which can be reacting with different types of antibody, different shapes, they actually will give a much stronger immune response. So the valence number is the epitopes on an antigen. Monovalent, one, multivalent, multiple. On the other hand, the antibody affinity is a strength with which the antibody binds to its antigen at a given antigen binding site. This is also important. On the other hand, this gives you an idea that sometimes some of the antigen are weakly bind to the antibodies, and these are considered as a weak immunogen. On the other hand, there could be a strong immunogen which can bind to the strong antibodies much more strongly. Another important is the avidity of the antibody is the overall ability to bind to the antigen at all binding site. What we mean by that, there is a subtle difference in between the antigenic determinant sites or the epitopes. Suppose you have a monovalent antibody which has a one binding epitope. So that doesn't even, you don't care about that at all because it does have only one binding site. But suppose you have an antigenic site which is triple valence, means there are three binding sites on that particular antigen. So that means that three binding site means there are three epitopes on that specific antigen. So the important point is that avidity of the antibody means that if all these three epitopes can bind to the three different antibodies at the same time, it does have a higher avidity of the antibody. On the other hand, what we mean by that, if there is one antibody once bound to that antigen blocks binding of the antigen to the two other epitope site, then the avidity of the antibody to that is less. So this is a very important thing, that doesn't matter whether you have multiple epitopes, because multiple epitope means that if you have, say, for example, you have epitope 1, 2, and 3. Some can bind to the epitope 1, some can bind to the epitope 2, and some can bind to the epitope 3. That is absolutely fine. But the problem is that if the antigen is of a shape where antibodies can bind to 1, 2, and 3, simultaneously 3 different antibodies, that antigen is higher in amount of giving immune reactions, and the affinity of the antibody is higher for that antigen. On the other hand, if it happens that if instead of 1, 2, and 3 different site, if an antibody binds to the site 1 epitope, due to the placement of the 2 and 3, no other antibody can bind to 2 and 3. Or say if an antibody binds to specifically to the epitope site 2, it will block the binding of other antibodies to 1 and 3. So depending upon that, the avidity of an antibody of an antigen also varies, whether it will allow all the epitopes to bind the antibodies or it will not allow to that. That has very different idea, that a valence number and the avidity. Valence number is the number of epitopes present on an antigen that can be recognized by individual antibodies. Avidity on the antibody is that at the same time, how many different antibodies can bind to the different epitopes present on the same antigen. So a very important concept. Then we are going to talk about the haptens, and these are small organic molecules, and they are by themselves not antigenic, but sometimes they become antigenic when bound to a larger carrier molecules, which are usually most of your own proteins. So for example, penicillin do not give any kind of reaction to most of the people, but there are some people who are allergic to penicillin. When we say that allergic to penicillin means that a person, if given injection of the penicillin or take oral penicillin, they will have an immune reaction, means that they will have an allergic reactions. An allergic reaction is an immune reaction. And why this will happen? Because for some particular patients who are allergic to that particular hapten here, that's penicillin, it will react to some of their skin proteins, and this is just their makeup of their skin proteins, slightly different from other people. And that will lead, once it binds to those specific type of proteins, will lead to a severe allergic immune reaction. There is a classic example for this, is that you have all heard about the poison ivy, and the poison ivy can give you really blistery bad kind of rashes. But the important thing is that a lot of people actually probably do not know that the first time you come in contact with the poison ivy, it really doesn't give you a very strong immune response. That you may get a little bit of reddening of your skin, but the first time if you contact poison ivy, it's not going to be a severe reaction. But what happens that it produces, due to your oxidation, once it goes inside your cell in the skin, due to the oxidation, it produces a quinol kind of molecule which binds, this is not necessary, this is just a little bit extra that's in your textbook as well, that these quinol type of molecule bind with some of your skin proteins and you get sensitization. That means your immune system do understand that this is a kind of a hapten attached with a carrier protein, and your body reacts to that. And that goes into your memory. That means that there are some memory cells already produced. They understand that this poison ivy quinol is a foreign substance and you have to react with that. So what happens that the second time when you touch the poison ivy, it's going to give you a severe reaction with the blisters in that area. Most of the times, that is unknowingly your second times you are touching the poison ivy. First time you just get sensitized by that very mild immune reaction, maybe just a reddening, but the second time or the third time, it could be severe. And this quinol molecule, which is present in that plant, in the poison ivy, is an example of hapten. It by itself, it is not an antigen, but it reacts or binds to one of your skin molecules, larger carrier molecule, and become an immunogenic. So the adaptive immunity can be earned or borrowed to some extent. We talked about this in the epidemiology chapter. So let's take a look at that. We have this naturally acquired adaptive immunity, naturally acquired passive immunity, or artificially acquired active immunity, which is vaccination, and artificially acquired passive immunity. Let's take a look at the picture, next slide, which will illustrate all these activities much more clearly. So the acquired immunity could be natural. So active immunity is the consequence of a person developing his or her own immune response to a microbe. When we get, particularly say for example, a disease, we naturally develop our own active immunity. Say for example, if a person gets a chicken pox, then they raise antibodies against the chicken pox naturally and they will be immune to it. On the other hand, natural passive immunity is the consequence of one person receiving preformed immunity made by another person. Classic example is the maternal antibody, and these antibodies are present in the breast milk. So what happens when a newborn immediately after birth do not have a strong immune system? It takes months to develop that strong immune system where all these natural B cells and the T cells are selected. And before the newborn can have his own immunity, the mother's immunoglobulins, which is present in the breast milk, actually protect that newborn child. And it is very interesting that there is a specific class of immunoglobulin, we are going to talk about that later, which is called the immunoglobulin G. That's an antibody. This is not degraded and it can pass through the intestine of that newborn. And there is a process called transcytosis where it is absorbed through the intestinal cells of the newborn. So these transcytosis means that these antibodies from the mom can get absorbed into the newborn's intestine, and from those cells, without getting degraded, they can be passed into the blood or the serum of the newborn. And that is the passive immunity, and that will protect the initial early months of the newborn from all the different pathogens. So both of these are natural immunities. On the other hand, what is artificial immunity is produced purposefully through the medical purposes, also called the immunization. So one of the artificial active immunity is the vaccination that we all know. So you make a vaccine, which could be a killed microbe or a part of the microbe, or a microbe which can elicit an immune response but will not cause a disease. All these different types of vaccine candidates that we talked about can be given to you, and you will have an immune reaction. But for the future, you will be protected because you already will develop the memory, which we talked about, which is the classic example of the acquired or the adaptive immunity. You will be protected for a future challenge by the original pathogen. The next is the passive immunity, is the consequence when a person receiving the preformed immunity made by another person. And immunoglobulin therapy is a very standard therapy in the cancer. So what happens, many of the cancer patients or the cancerous cells will produce some of the very specific type of protein on their cells which are not natural. That means that your cancer cells have a specific type of antigens which are not present in other type of your cell. And there are many specific type of antibodies. Usually these are most monoclonal antibodies, very specific to that type of cancer. And that can be given to you as a immunoglobulin therapy or to the patients, which can lead to the remission of the cancer. There are some very good immunoglobulin therapy for some particular cancer. But again, remember that it does not work for all the different types of cancer. It just depends what type of cancer they are. And if there is a particular mutation in that cancer, which produces a protein very different from your own natural self antigens or your self proteins. That is important. Another very important aspect of the passive immunity is the antivenom or antitoxin. Think about that when you have a snake bite, like particularly a poisonous snake bite, you are rushed to the hospital and you are given a shot, which is an antivenom. These antivenoms are nothing but immunoglobulin shots. So how it is done? So from all these venomous snakes or other different sources of any of these toxins, these small toxins are injected into different mammals. And usually these mammals in the laboratory or in the industry, this could be goats, this could be even sometimes horses, this could be cattle. And those are maintained and raise antibodies. And these antibodies from these animals, like see these goats and the horses, they will be time to time humanly bled, and the serum is isolated from these animals. And then the immunoglobulin from these mammals are isolated and packaged. So these are the specific immunoglobulin. This comes in to the patients as an immunoglobulin therapy. Why this works? Because remember that most of the mammals do have these immune system which are very similar. So our antibody structures among the mammals are very similar. So think about this, that suppose you are working on a viper or a rattlesnake venom. So this rattlesnake venom is isolated, and then a small amount of rattlesnake venom is injected to a goat in an industry where they maintain these antibodies. That small amount of venom is good enough to elicit an antibody, but it will not kill the goat because the toxin is too less. But that toxin is good enough to raise antibodies against it into the goat. And they are given booster doses time to time. And then the goat will be bled humanely to some extent, as if it's donating blood. And from that blood, the serum will be isolated. From the serum, that specific antibody will be isolated. There are some specific ways to do that, which is called the antibody affinity purification, using that toxin back again. And these affinity purified antibodies will be packaged, and these are present in the medical market to give it to you if there is a need. And these are all examples of your passive immunity. So the next part is that we're going to talk about the recognition of the foreignness is critical for a strong defense. And we are going to compare that how the cell receptor distinguish self from non-self, and compare the classes of these MHC class 1 and 2. These are the major histocompatibility complex which present these different foreign material to the immune system or the adaptive immune system. We slightly touched that in our discussion in the innate immunity, but now we are going to talk about that in detail, and you will be able to correlate these two things. And also we are going to identify the cells that function as antigen presenting cells. We also talked about that. Your macrophages are antigen presenting cells. Your dendritic cells are antigen presenting cells. Even your neutrophil can present antigen, so as your normal bodily cells. We're going to talk about that. So the immune system must recognize the foreign antigen as non-self and recognize host cells as self. This allows for selective destruction of the invading pathogens without destruction of your host tissues. We already talked about that this is the general idea, and this involves major histocompatibility complex. So what are those? Major histocompatibility complex, or MHC, and why they are named like that. So these are collection of genes which code for self or non-self potential of a vertebrate. So in humans, these are located on the chromosome 6 called human leukocyte antigen complex, or HLA, which gives rise to these MHCs. And these MHCs, all these classes, the MHC 1 molecules we are going to see, these MHC class 1 molecules are found in all types of nucleated cell. All types of nucleated cell. They're important for organ transplantation. Why this is so? So this class 1 molecule of the MHC, which found in all type of nucleated cells, they have a class A, B, and C types. And what you get one set of ABCs from your mom, one set from your dad. And as you get those specific ABC types, you have a tissue specificity of the class 1 MHC molecules. So this is individual polymorphism that exists individually. So a related person that your family members will, like your grandparents, then your parents, then your siblings or your child, will be closely related with these MHC molecules. That's the reason sometimes you will see that in the organ transplantation, there are some great stories where a family member donates an organ to a person in the family, because what happens that this is the basis of the tissue match or rejection. So all you know that a person need a heart transplant or a liver transplant or a kidney transplant, and they wait and wait and wait until they get a tissue match. What do we mean by that tissue match? That in between the donor and the patient, these class 1 MHC molecules, ABC types, should match very closely. If it does not match, your immune cells will attack that transplant and reject that transplant as foreign. And this is the basis of the important for organ transplantation. Similarly, on the other hand, there is a class 2 molecule which is only present on the cell that can process and present antigen to T lymphocyte. So they are present on the APCs. Mainly, these are your immune cells, which are the macrophages, phagocytic cells, dendritic cells. These are antigen presenting cells, and also B cells. So you can see that the class 2 molecules are found on very specific immune cells, which present the antigen to the immune system. They are not found anywhere else. And then there's a class 3 molecule that is not needed, includes secreted proteins, not required for self, non-self, include various secreted proteins, but that is beyond our scope. So we are only interested of MHC class 1 and class 2. So let's take a look at this structure. So MHC class 1 do contain a molecule which is larger. This is a heterodimer of an alpha protein and a beta protein. So this alpha is about 45 kilodalton and do contain this alpha 1, alpha 2, and the alpha 3 domain. You can easily see. And then there is a transmembrane domain, and there is a cytoplasmic portion of this long molecule, about 45 kilodalton in size. You have a much smaller, which is known as a microglobulin. This is just only a 12 kilodalton, and this is the other protein. So you have one of the monomer, that is the alpha protein, and the second beta 2 microglobulin is the other protein, so alpha and beta. So they sit outside the plasma membrane, and in between the alpha 1 and the alpha 2, there is a cleft, which is the antigen binding site showed in here in the space filling model. So here, the MHC molecule or MHC1 will load an antigen to be seen by the immune system. And we'll come back to that, the detail about that in a second. The second thing that you have another thing, which is the MHC2. This is also a heterodimer with two distinct proteins. And each of these dimers do have an alpha-1, alpha-2 component, transmembrane domain, then a short cytoplasmic domain. And also you have a beta-1 and beta-2 domain, alpha-1, alpha-2. This is your alpha protein, and a beta-1, beta-2. So this is another domain. But they are very distinct, the MHC1 and MHC2. What you are seeing right here, you have a cleft where there is an antigen bound to this alpha-1 chain, alpha chain, and the beta chain, which is the alpha chain and the beta chain. So what is the difference in between that? From the structure, you can see that this does have an open-ended structure, whereas this is very tight. So what happens in the real three-dimensional structure, the MHC1 molecules are able to show only small and short peptides. So the antigen which are presented by the MHC1 molecules are much smaller compared to a much larger antigen can be presented by the MHC2 molecule. And we will see that this will allow large non-self antigen to be presented by the MHC2 molecules, whereas the MHC class molecule always present the self as well as some non-self molecule. And this is important because why this is done, think about that as your immune cells are being developed, all nucleated cells do contain these MHC1s. Remember that. We talked about that in a second. All nucleated cells do have these MHC class 1. So what happens that all your proteins in every type of cell, they undergo degradation in the proteasome as a part of a natural recycling of your proteins. So as your proteins gets recycled in the proteasome, small fragments of your proteins are loaded onto the MHC1s, and these cells are present throughout your body. So what happens that when these cells present these antigens to the developing B cells or the T cells, as they are loaded onto the MHC class 1, they understand that probably this is a self antigen and kind of flags it that I am self, do not touch me. On the other hand, there are some cases that it can show a foreign antigen as well. We are going to see that. On the other hand, the MHC class 2 will never present the immune system with a self antigen. Anything that is presented by the MHC class 2 will be always foreign. So again, MHC class 1 can present either self or foreign, and we'll see that if it presents foreign, what happens to that cell, whereas the MHC2 will present always a foreign antigen. We'll come back to that in a little bit more detail. So right here, that leads to two things. One, what is called as endogenous antigen processing, and another is an exogenous antigen processing. So what is the difference? We already talked about that. The class 1 binds to the antigen peptides that originate in the cytoplasm as the action of the proteasome and present the antigen to the CD8 plus cells. So what happens, there are two things can happen, and we are again going to talk about that what is CD8 plus cells, because we still do not know this. CD8 plus cell is a cytotoxic T cell. So once your MHC1, which is present in all of your cells, nucleated cell, when it presents the antigen to the cytotoxic T cell, it will recognize your antigen. But on the other hand, to activate that cytotoxic T cells, you need another stimulus, and that stimulus is not present in your own cell or your self cell. That's why the CD8 T cells, which is a cytotoxic T cells, will leave your own cells alone. So no matter what, it will leave yourself because you presented that with MHC1, which is recognized by the CD8 cytotoxic T cells because you have an ID, and it left you alone because you do not have an additional particular co-receptors that is present on your cells. On the other hand, see that what happens with the exogenous antigen processing. And we'll explain that a little bit better because it may sound a little bit grayish and may not completely make sense here because we are missing some of the more information that is required to understand this fully. The next part is the exogenous antigen processing, class 2 antigen fragments that come from outside the cell. These are foreign. These are present to CD4 helper cells. And here what you are seeing, that suppose this is a dendritic cells or which captured the pathogen by endocytosis, or this is a macrophage which captured the pathogen by endocytosis, or a neutrophil by phagocytosis. All these, once they are degraded inside the endosome by the help of the lysosome, from these endosomes, they are going to go into the endoplasmic reticulum loaded onto the MHC2, and these MHC2s will be directed towards the cell surface from these endosomes. So right here, you can see that these are the digested fragments of the pathogen, which contains a foreign antigen loaded onto the MHC2, and now this MHC2 is displaying that processed peptide outside the cell surface. Now, this will recognize the CD4 T helper cells. So these CD4 T helper cells do have this cluster of differentiation 4. We are going to come back to that. And these CD4 T helper cells will only recognize the pathogen presented with the MHC2 molecules. So these MHC2 molecules are recognized by the CD4 or the T helper cells, and they will activate the immune system. So that is the most important part. So there are two things that you definitely have to understand from here. If there is a MHC1 presenting an antigen outside the cell, it is recognized by the CD8 cells or the cytotoxic T cells. So remember a number here, 18, class 1 and CD8, 1 and 8. 18 is another one number, another is 24, MHC class 2, recognized by the CD4. So 2, 4 and 1, 8. That's the combinations. Class 1 MHC recognized by the CD8 T cells. Class 2 MHC is recognized by the CD4 T cells. So what happens, the class 1, once it's recognized, as I said, that if it is a self-antigen, CD8 will leave it alone. But on the other hand, say for example, if that particular antigen is a non-self antigen, then the CD8 cell will kill that cell along with the pathogen. But how will the CD8 cell will know that it is a foreign antigen? Because I say that when you present that with an MHC1 molecule, there is a lack of a particular co-receptor that is present not on the surface of these cells. So here it comes into action that suppose you have a pathogen inside your body which is infecting your cells, suppose a virus, and that virus is also taken up by the dendritic cells or the macrophages, ingested, and that part of the virus is now shown outside by the MHC class 2. MHC class 2 is showing that virus part. Now this CD4 T helper cell will trigger the immune cells, and it will arm the cells, or the CD8 cytotoxic T cell will be presented by these CD4 T cells to activate themselves. Now the CD8 T cells know that, okay, all right, this is a virus, this is an antigen which is non-self, and if I find a cell presenting me with that specific virus, I need to kill that cell. That's the trigger. So what is happening that when you have your self proteins shown by the MHC1 to CD8, there is no CD4 activation, no co-receptors, and that will leave your own cells alone by the CD8. But if it is a pathogen and your CD4 T cells trigger your immune system, trigger your CD8 T cells, which recognize these exogenous pathogen, it will trigger the immune reaction in the sense that it will kill the cells because it will understand that this cell is now infected by that virus. That's the reason that this cell is showing me endogenously processed epitope of the foreign pathogen. So let's take about the critical cells of the T cells. So the T cell development is a multiple subset of the T cells work together to initiate, orchestrate, and carry out adaptive immune response originated from the CLP. And there is a thymic selection. And in the thymic selection, we already talked about that. In the thymus, this is a gland just above your heart, which determines what kind of T cells any given immature cell will become. And this is defined by the structure of the T cell receptor and then by the structure of the T cell receptors, and also determines the co-receptor specificity. And what we mean by that? There are some specific cell which we call as an alpha and beta T cells. These are the higher in number, but there are some small portion of cells which are known as a gamma delta T cells. They have a different type of receptors, and most of these gamma delta T cells from the thymus, about 2% of the total T cells are these gamma delta T cells. They will leave the thymus and they will mainly go into the skin and in the mucous membrane. On the other hand, your alpha beta T cells will go into the other areas and particularly into your lymphoid tissues and the lymphoid organs. Now, why you have these particular gamma delta T cells? Gamma delta T cells are more diversified, and as they go to your first line of defense in the skin and the mucus, their combination of their receptors are much more diverse to see or bind to much more variety of different types of antigens. So their diversity to bind to the antigen is much more than the alpha beta cells. And that makes sense because these gamma delta T cells are at the skin or at the mucous membrane, which are basically the areas through which a pathogen will gain entry mainly into your body. Also, it will determine the co-receptor specificity. So once these alpha beta T cells do have that specificity, then they will be added with the specificity of either CD4 or the CD8. The CD4 becomes the T helper cells, and then they will have T helper 1, 2, 3, 4 different types, and also the CD8, which will be the cytotoxic T cells. And then they will go into a specific destination. And the T cell co-receptors, on the other hand, are the members of the family of cluster of differentiation, or the CD molecule. And there are additional CD molecules, addition to this CD8 and the CD4, and we will see that this is also important. There's a different molecule called CD28 that is required also as an additional signal for the activation of the T cell. Activation, not the development, activation of the T cell, and we're going to talk about that a little later. So here we are looking at the development function of the B and the T cells. We are first only going to look at the T cells, and we will come back to the same diagram when we talk about the development of the B cell. Here we are looking at the common lymphoid progenitor cell, which mature in the thymus into the T cell. So this T cell will have its own clone. Clone means that the T cell receptors in these cells will be the same as well. Then these T cells will differentiate into a TH cells, which is a T helper cells, and growth differentiation factor. At this point, what happens that these T cells, the antigen stimulus is required at this time. So at this point, due to the antigen stimulus, two things can happen. This antigen stimulus could be the self. If it is a self-antigen, and if your T cell receptor reacts with that, those cells will be immediately killed. That means that it will not do anything. On the other hand, if it doesn't react with any of your self-antigen, it will be chosen and it will wait there until there is an antigen stimulus, and then it will differentiate into a T helper cell. This will release growth differentiation factor. This will enhance or suppress the immune cell action, including the B cells. And some of these T helper cells will become memory T helper cells and will remain in your system for the rest of your life mostly. And that's your T helper cell development. These contain the CD4. It's cluster of differentiation D, CD4 on their surface. On the other hand, another type of T cell which differentiate into the cytotoxic T cells, CTLs, they do have CD8 on their surface. Upon antigen stimulus, this will again release lytic enzymes and the proteins, which will kill the altered or infected cells. Altered cell means, think about a cancer cell, which can have a particular antigen on their surface or a protein which mutated and is in different form. So the cytotoxic T cell can kill them as well. Or infected cell, as we talked about that. And some of these cytotoxic T cell will become memory T cells. But to kill these particular cells of yours, it needs a stimulus from the helper cells. And we're going to talk about that particular actions, that how this happens. So here we are looking at that what are the T cell receptors or the TCRs. This is very important. Remember that we already talked about that what presents the T cell receptors with the antigen. Remember that that is a separate thing, which is a major histocompatibility complex 1 and 2. Those are the specific type of structures which will present the antigen to the T cell receptors. So they reside in the plasma membrane, and you can see that this is the structure. They do have 2 different individual monomers, and these monomers, so the T cell receptor is a dimer, and it contains a variable alpha and a constant alpha chain, and it also contains a variable beta and a constant beta chain. What do we mean by that? The constant portion is the same in between or among all the T cell receptors. What changes is the structure of the V alpha, variable alpha, and the variable beta. So this changes, and this antigen binding groove gives a huge amount of diversity to these cells. Whether it is alpha, beta, or gamma, delta T cells, it can be a very large number of combinations, literally in trillions, to bind any kind of three-dimensional foreign molecule structures. So that's your T cell receptor. In addition to that, it also needs, the whole T cell receptor needs some additional structures which are known as these ITAMs, which are basically the immunoreceptor tyrosine-based activation motifs. And these ITAMs are also known as the CD3s. And these are the core receptors, these CD3s, ITAMs. Why these are required? If you carefully look at that the transmembrane domain and then the C-terminal domain of the T cell receptor, the real T cell receptor, is very small, and this cannot lead to much of the signal transduction. So what happens? These CD3 molecules, which are the additional C3 molecules, you can see that there are different ones. And they are all different types. You can see that they are delta epsilon, they have a gamma epsilon, they have tau, they have all different types of combinations, do not need. But these CD3 molecules or the ITAM will cause a signal transduction going on, which ultimately will lead to transcription of specific genes inside the cell, which will lead to, again, a large number of proteins to be expressed by the cell or secreted by the cell, including the cytokines. That is the activation of the T cell, and which will lead to activation of many different things in your adaptive immune response that we are going to see that. So here in this picture has been shown that a dendritic cell, which is an antigen presenting cell, which will have a major histocompatibility complex MHC2 molecule, in addition to MHC1 molecule. Remember that MHC1 molecule is present in all nucleated cells. So an antigen presenting cell will also have an MHC1, but this also contains the MHC2, which is not present in a normal nucleated cell, which are not macrophage or not B cell or not dendritic cells. The MHC2 is not present other than those cells. So here you are seeing what is called as an exogenous antigen presentation. Means this dendritic cells caught that antigen in the fragment form or in a large form. Suppose it's a pathogen that infected a particular host. That pathogen is internalized by the process of phagocytosis by the dendritic cells, degraded, and then shown or loaded onto the MHC class 2 to show it outside the plasma membrane. Now this, which is shown by the major histocompatibility complex, is now attached to a TCR. And once the TCR get activated right here, it will make some conformational changes to these co-receptors or the CD3's items. See that they are close, but once you have that antigen bound to the T cell receptor, they are now all close and attached to this C alpha and the C beta, constant alpha and the constant beta part. They're clustered together. This will cause this intracellular signaling to turn on expression of certain genes. And there are thousands of different genes that could be switched on due to this interaction. So here we are looking at that the T cell activation requires binding of specific antigen, and there are some very specific type of interaction that needed to be taken place so that there is nothing wrong that goes on. So there are several tiers of specificity. So here we are looking at that this is an antigen presenting cell. This is a dendritic cell. Right here, your T cell receptor is recognizing. Remember that it will also have the items right here, which is not shown in the figure. That's there. But now see that there is a very specific thing. This particular T cell do contain a CD4 co-receptor. So this CD4 co-receptor is needed to activate this particular cell. So here you have an MHC class 2 and you have a CD4 molecule recognizing that, and this will activate. So the CD4 is along with your T cell receptor, all your items are the CD3. CD4 is required. So 2 and 4, remember that? That will activate this T cell. There is additional signal required. A second signal is required for the lymphocyte proliferation, differentiation, and expression of cytokine genes, which is presented by this B7 or CD80. So this CD80 molecule do have this particular signal, which will be recognized by this CD28, and all T cells will have this CD28 molecule. So once this B7 or CD80 is presented, it will activate the T helper cells. Now, let's see what happens in a different case. So first occurs through the antigen presenting bridges of the MHC class 2 to the APC2 of the cell receptor. This is known as the immune synapse. So this is your immune synapse. This will initiate signaling cascade involving other membrane-bound proteins and intracellular messengers. And the second signal is required for the lymphocyte proliferation, differentiation, and expression of the specific cytokine genes, which is your second receptors right here. This is through your B7 and CD80. That's very important. So let's see that what is happening in a different case. In this case, what you are seeing, still, this is an antigen presenting cell right here, antigen presenting cell. As I said, that all nucleated cells in the body will have MHC1. So here you have the same, say suppose the same antigen presenting cell or the dendritic cell, but here the antigen is loaded with this signal, which is specifically loaded onto this MHC class 1. And now this particular cell is now contacted with this CD8 or the cytotoxic T cell right here. So what is the difference in between this and this? We're going to come to that. So here you can see that this is your B7 or the CD80 molecule that will also activate the CD28. So what is the difference? You can easily see that even if you have an MHC class 2, which will activate downstream differentiation expression of the specific cytokine genes, but this B7 or CD80 is not present. Make sure that you understand clearly. This B7 is CD80 is not present in the normal cells. And that's the reason that a normal cell will not be able to activate the lymphocyte proliferation, differentiation, or expression of the specific cytokine. So what happens that the CD8 will recognize that there is a foreign antigen, it will get activated and kill that cell, but it will not be able to activate the immune system. On the other hand, to activate the immune system, you need a presentation of the MHC class 2 or MHC class 1, no matter what, but you need the B7 or CD80 in additional signal, which is only present on antigen presenting cells, but not your normal nucleated cells. So antigen presenting cells can trigger the immune response, the second signal, or the cytokine response and the proliferation, but not a normal cell. On the other hand, the third signal that is also important is the release of the cytokine by the antigen presenting cells, which is not shown in here. So along with this activation, the antigen presenting cells, the dendritic cells or the macrophages, will also release cytokines that will act on the lymphocyte as a third signal. So all these three signals are required for these specific ones, and some of these will differentiate these T cells into memory and effector T cells. So the take-home message from here is that once a T cell is presented by an antigen, it will become an active or effector T cells. So remember that once the T cell develops into the thymus, it is already a cell which is an developed T cells. It's a developed T cells, but it is ready to go, but it has not seen any antigen yet. Once an antigen is seen by that developed T cells, it will transform by these additional signals into an effector T cells, which will spring into action to activate the immune system. So that's the nomenclature. So let's take a look here. So the mature T cells, which I explained, are naive until activated by the antigen presentation. Once activated, that just we discussed, proliferate into effector T cells and memory C cells. And the effector T cells will carry out specific functions to protect the host against a foreign antigen. And they could be three types: T helper, then they have T regulatory cells and cytotoxic T lymphocytes. The T helpers do have the CD4s. The cytotoxic T lymphocyte, or the CTLs, they have the CD8s. So let's see T cell progenitor. This differentiate into the thymus, into the T regulatory cells, alpha beta cells, we talked about that, or the gamma delta T cells. So what are these things do? That the T regulatory cells maintain the homeostasis by secreting cytokines that downregulate strong immune response to non-self. Mainly, these cytokines are IL-9, IL-10, and TGF-beta. And you will see that I will mention some of these particular ILs or the interleukins and the different cytokines. But again, the detailed memorization of each of the cytokine, what it does, it's not necessary. Just remember the overall function with that. But what is important that sometimes this is very important. Non-self strong immune response against the non-self is suppressed by the T regulatory cells. And again, remember that we talked about this super antigen, and this super antigen binds in a certain specific way to give 1,000-fold increase into the activity. And sometimes at that time, even these T regulatory cells fail to stop that immune response. On the other hand, for most of the times, there is a fine balance of the homeostasis, and these T regulatory cells make sure that your immune system is not overreacting. It is reacting appropriately, but not too much, because too much activation of the immune system will have a deleterious effect on your body. Your body can go into an inflammation, into fever, and these can have detrimental effects in your body other than killing the pathogen. So T regulatory cells do that. Alpha beta T cells, majority of the CD4 and the T cells are this type, exhibit the helpers and the cytotoxic function. Memory cells arise after they are activated. On the other hand, the gamma delta T cells is a small subset, less than 2%, found in the gut mucosa and the skin. And again, as I said, respond to a greater diversity of the antigen than the alpha beta cells, because they are exposed to the first line or the front line where they can see much more pathogens compared to the alpha beta T cells, which are in the internal body compartment. So what are the different ones? So we will come to this in much more detail, because each one of them actually do have much more functions than it's written in here. So we're just skimming here just for a second. The T helper cells overall traits, known as a CD4 T cells, activated by the antigen presentation by the class 2 molecules, so 2,4. Most important types of helper cells, Th0. These are the mature, naive T cells, not yet activated. They're just waiting for antigen to be presented. So once they're activated, they will differentiate into the Th1. This will help to activate macrophages, but there are other functions. These are just some of the basic functions. They will activate B cells and other, and we'll come back to that. Th2, on the other hand, help produce B cells, some antibodies. Th17 assist for antibacterial responses, and T regulatory help to control the lymphocyte responses. These are some of the primary function, but there will be much more detailed secondary function, and we're going to talk about that in a minute. So the next one, other than the CD4, which is the helper T cell, is the cytotoxic T lymphocyte. These are CD8. Remember that? They will recognize the MHC1 molecule, and these are 1818. Once activated, the CTLs can kill host cells that have been infected by intracellular pathogens, such as a virus, are presenting cancer neoantigens, or have some antigen MHC1 combination that originally activated the CTL. So after binding the target, the cytotoxic T lymphocyte will kill the target via this perforin pathway in an apoptotic programmed cell death pathway. And we're going to discuss that in a second as well. So here we have a little bit more detail about the T cell development and activation. So although it is written here as development 2, but I would like to say that T cells are already developed in the thymus, and here we are seeing the activation of the T cells. So let's take a look one at a time. So here we are looking at that a dendritic cell's antigen presenting cell is presenting an antigen with the MHC2, and here you have a Th0, which is the naive T cells with the CD4, and it will have the TCR or the T cell receptors with the CD3. Those are the normal, and the CD4 is the co-receptor. Once it sees the antigen presented by the MHC2, this Th0 will get activated and will become an effector T cells. And these are the effector T cells, and see that what it will do. So the first thing what it will do, that let's go one at a time, that these T helper cells can differentiate into by the production of IL-12 and IFN gamma. So if the CD4 T cells release IL-12 or IFN gamma, it's going to transform the Th1 type T helper cells. So what's the Th1 type of T helper cells is going to do? That it's going to release interleukin-2, tumor necrosis factor, TNF, and interferon gamma. And it's going to activate the CD8 T cells. And there are some specific ways that, again, not necessary, but it will specifically works with a transcription factor, which is called the T-bet. And this T-bet, the T-bet is a transcription factor which will allow synthesis of this IL-2, TNF, and IFN gamma to be released from the Th cells to activate the CD8 or the cytotoxic T cells. Next one. So after 1, let's go to the 2. So IL-4 released by this will activate the Th2 cells. And how it does? IL-4 will activate a transcription factor which is known as a GATA3. This GATA3 will activate and the transcription factors which will activate the secretion of IL-4, 5, 6, 13, which will activate the B cells. On the other hand, see that what happens right here to once the CD4 T cells release the TGF-beta, IL-6, it will activate the Th7, and here there is a particular transcription factors which is known as the ROR gamma T, which will help the IL-17 to be released to result recruitment of the neutrophils, which are the phagocytic cells. So you can see that how this activation happens once the CD4 Th0 cells bind to the MHC class 2 presenting an antigen, the T cell receptors will make changes into the cell because there will be some interaction going on with the T cell receptors with the ITAMs or the CD3. As I said, that it will induce some of the signals, and these signals means that these T helper cells will release specifically all these different cytokines. These cytokines will specifically act on specific type of transcription factors, and these transcription factors will lead to development of these T cells. So say for example TGF beta, and this TGF beta will again work on these CD4 regulatory T cells, and they work with this specific type called a FOXP transcription factor. So TGF beta works on that transcription factor. That will again induce the IL-10 and the TGF beta production in larger amount, which will suppress the immune function. So this is a one way of activation of the adaptive immune system. Let's see that what happens if a dendritic cell is presenting the antigen with an MHC class 1. We saw the activation by the MHC class 2. See what happens with the MHC 1. Now, MHC 1 is activated by the CD8 or the cytotoxic T cells. It can convert into a memory T cells. It can be converted into a cytotoxic T cells. It can convert into a cytotoxic T cells regulatory cells. So this cytotoxic T cell again can differentiate, or it will make its own clone to attack the cells. So here there's a very important aspect. Remember that. So why the CD8 cell is not killing the dendritic cell? That is very important. Why it will again understand an MHC1 presented antigen by a normal cell, and here the cytotoxic T cell is killing the normal cell death by apoptosis. Here the dendritic cell also presented the antigen by an MHC1. The cytotoxic T cell is not killing the dendritic cells, but it's killing the normal cell. And why does this happen? Let's scoot back a couple of slides to understand that why this difference happens. So right here, if we look at that, right here we are seeing that this is an antigen presenting dendritic cells, which does have the MHC class 1 presented to the T cell receptor. See that there is an additional molecule, which is the B7 or CD80, which is recognized by the CD28 receptor. This is a signal, or this B7 or CD80 is only present on antigen presenting cells. Once it has the CD80 presented, the cytotoxic T cell understand that this is an antigen presenting cell. This is not a normal cell. So it is okay that if MHC1 presents the antigen, because it just got that from the cell itself, because it is a antigen presenting cell. But the difference is that this CD80 or B7 is only present on the antigen presenting cells, not in other cells. Your other regular cells will not have the CD80 or B7. So what happens here? As your own cells do not have the CD80 or the B7, it will not be recognized by the CD28. It is only presented by the MHC1, and immediately it understands, okay, this is not an antigen presenting cell. It is a cell that is infected by this pathogen, and it will kill it. And the process is that it's going to release these perforin, granzymes, granulysins. These are some of the molecules which will lead to the apoptotic cell death. And also these cells do have what is called a Fas ligand. These Fas ligands are specific death or apoptotic receptors, and these are presented on the CD95 on the target cells. And these apoptotic receptors will receive specific signal from the cytotoxic T cells to kill the cell by apoptosis. So this is how an infected cells can be killed by this cytotoxic T cell. On the other hand, by the presentation of the antigen, you can see that B cell is going to get activated, more neutrophil is going to activated, and that how this whole immune system is now orchestrated. The next part, what we are going to see, that you have seen that the presentation of the antigen is activates the B cell to produce the antibody. We are going to see that how the B cell make antibodies. Describe the B cell receptor structures, function, what they are going to do, and how the steps required for a B cell to become an antigen presenting cell, and compare the T and dependent and cell independent activation as well. So we are seeing this diagram once again. We already saw the T cell development, here we are seeing the B cell development. So again, the common lymphoid progenitor cell, CLP, mature in the bone marrow, and the B cell clones will be produced. Once it gets the antigen stimulus, it can go into two things. It can differentiate into a specific type of even larger cell, which we call as a plasma cell, which is going to secrete the antibody to neutralize the toxin, viruses, and opsonize the bacteria, which will also trigger, remember that, the complement system. On the other hand, some of these cells will become the memory B cells, and this will initiate the rapid response to reinfection with the same antigen. So let's take a look at what are the downstream process. So the first thing in the B cell biology, we are going to see that what are the different types of receptor that are present on the B cell surface. So we already now know that you have the TCR or the T cell receptors that bind to an antigen. On the other hand, here we are looking at the B cell receptor. So the B cell must be activated by a specific antigen, and cells then replicate to differentiate into the plasma cells which secrete antibodies. So how these receptors work? The B cells have an immunoglobulin receptors called B cell receptor for specific antigen that will activate the particular B cells. An interaction with an antigen is communicated to the nucleus via signal transduction pathway, similar to the described to the T cells, which will regulated by these ITAMs or the CD3 co-receptors. But one of the important thing that remember that what we are looking at, that this is a B cell receptor complex, and this is an immunoglobulin M-type molecule in its monomer. What we mean by that, it may sound a little bit weird at this point. IgM is an antibody that is present in your serum as well, which is pentameric with 5 of these similar structure. 5 of these. It's like a pinwheel-like structure. So instead of 5 of these, as a B cell receptor, you have 1 copy of this as a receptor. And you have 1 copy of this IgD. This is a different class of immunoglobulin. This is present. So you have 1 monomeric IgM, 1 monomeric IgD, along with the CD3 co-receptors. So what they have, on the other hand, each one of them do have 2 chains. If you carefully look at that, this is a short chain. This is an individual polypeptide chain. You have a short chain right here. You have also a short chain. So you have 1 short chain, another short chain. That means you have 2 short chains. So these are a little bit smaller in size. On the other hand, you are seeing that right here, this is a long chain or a heavy chain. This is a long chain or a heavy chain. So if you look at that, here it is written as V, H, and L, and we'll explain that what it is. Remember that V, H, and V stands for variable. So variable means this blue part right here, the blue part for all of these is the variable region of the B cell receptor. That means that this is the epitope binding sites. So these are the diversified with billions and millions of these combinations. So these vary among the receptors of the individual B cell clones. On the other hand, this yellow part for both of them is the constant part. On the other hand, what is being shown, the variable part have a variable heavy. That means this is the variable heavy chain, H, and a variable light chain, that is L. So variable heavy, variable light, variable heavy, variable light, the same thing. That is meant by the VHL. On the other hand, you can think that this part is constant heavy and this part is constant light. You can tell that this is Cl and this is Ch, constant heavy and constant light. So you have 4 polypeptides and these are all connected to each other, disulfide bridges, so that they are kept together. Then the final thing is important. See that this is very interesting. The B cell receptor complex is made up of 1 IgM, 1 IgD. One epitope can bind here, another epitope can bind here. So it can simultaneously bind 2 antigens. Remember that. It can bind 2 antigens. This is different from the T cell receptor. If we just go back to the T cell receptor, it can bind to only 1 antigen at a time. Very different. T cell receptor bind 1 antigen at a time, whereas the B cell can bind to two antigens at a time due to the structure. So there are two mechanisms of the B cell activation. One is called a T cell dependent activation, involves the interaction with the T cell. On the other hand, there is a T independent activation, which is triggered by the B cells to produce antibodies without the T cell. So how it happens? In the T cell independent cell activation, usually a polymeric antigen with large number of identical epitopes, say for example, bacterial lipopolysaccharides or peptidoglycan. You can see that this is an B cell. All these are these two of these. So these are all different receptors. Suppose instead of binding just to one IgD and one IgM molecule, if there's a large antigen which is covering multiple of these receptors, that will activate the T cell independent B cell activation. But here the problem is that antibodies will have a low affinity for the antigen. And in this case, no memory B cells will be formed. That is one of the problem of the independent B cell activation, not the most efficient way for activation of the B cell. On the other hand, what happens that the T cell dependent B cell activation involves interaction with the T cell, which gives a much more stronger response. So why this is even exist? Think about that. You just got an infection, but it did not, or the bacterial infection could not even pass. We should not even say that bacterial infection. Say there is a bacterial invasion to your cells, and it did not even pass through your innate immune response. That means that it just was taken care of by the neutrophil and the macrophages. But sometimes you know already that the neutrophils will release some of these fragments of the digested phagocytic product, and your macrophages will do that. Suppose they release in the tissue space or in the blood plasma some of these bacterial lipopolysaccharide or bacterial peptidoglycan, which are the residual part from already digested or killed bacteria, not a real threat. At that time, the T cell independent B cell activation can happen to clear them out. And if there is low affinity for the antigen, no memory B cell is formed, there is no problem with the system. On the other hand, so this is activated mainly by the residual amount of antigen from your innate immune response, which took care of that particular invasion, and there is no real reason to activate the adaptive immune response. And this happens to your body every day, all the time, simultaneously, without any kind of adaptive immune response. On the other hand, when the T cell dependent B cell activation means here that your innate immune response could not stop the invasion of the pathogen or the toxin or the product. And at this time, you need the effect of the adaptive immune system to kick in, which is a much more stronger response. So like T cells, the B cells will also require two signals. One is antigen B cell receptor specific interaction, and also activated T helper cells too, which binds to the B cell, present the antigen, and secretes the B cell growth factor. That B cell will differentiate in the plasma cell, in the memory cell. So if the T helper cell is not involved, then there will be much less activity of the cell. So let's see what is happening. The whole thing is now much easier. This is your dendritic cells, which is presenting by the MHC class II molecule, an antigen, to a naive T helper cell, Th0. Upon presentation, this will again convert into an effector T helper cells, and it is releasing IL-2 by itself and activating itself. These activated T helper Th2 type 2 from Th0 to Th2 by the selection expansion will now activate a B cell. This is a signal one for the B cell, which binds to the antigen. But along with that, to really proliferate, you need this second signal as well. So that's your second signal. So the B cell proliferation and the differentiation is due to the B cell growth factors, which can include the IL-4, 5, 9, 13, and IL-1 from even the macrophages, which is not shown. And all these, along with this IL-1, IL-6, there are many different once, all these cytokines will work on the B cell and they will transform the B cells into the antibody secreting plasma cells. And particularly for the plasma cells, the IL-2, IL-4, IL-6, and IL-13, those are very important for the plasma cells. Much larger with very, very rapid protein synthesis, which produce this immunoglobulin molecule. But what is the difference? A regular B cell will have these B cell receptors onto the surface of the plasma membrane to recognize the antigens, and also the B cell is going to get activated by the Th2 helper cells. Now, once the B cell goes into the plasma cell or this condition, it will now start secreting the antibodies from the B cells, and this will go into the blood plasma, in the serum, in the tissue space, and do. So we are now looking at the antibody and its development. So these antibodies is an immunoglobulin or the Ig. This is a glycoprotein made by the activated B cells, which we call as a plasma cells. And this serves as antigen receptors or the B cell receptors on the B cell surface. These are found in the blood serum, tissue fluids, mucosal surface of the vertebrate animals. An antibody can recognize and bind antigen that causes its production as well. So these are some of the generalized idea about the antibody. So let's take a look at these structures. We already talked about that. It contains these 4 polypeptide chains, 2 identical heavy chains. So 2 identical heavy chains right here and 2 identical light chains. And there are some names. This portion is known as fragment AB, fragment AB. This portion is known as fragment C or constant as such. Both chains are connected, the heavy and the light chains, connected by these disulfide bonds, and these are the disulfide bonds in the red. You can easily see that. And also both chains contain 2 different regions, a constant C region, which is constant light and heavy. And again, if we just see that, this is your constant light and heavy. And right here, if you carefully look at that, this is your constant heavy, constant heavy. This is your constant light, constant light. This is your constant light. This is your constant heavy. So all the blue parts are the variable regions that we talked about, that V regions, variable, and the blue regions are variable light on the light chain. And this is the constant light on the light chain. On the heavy chain, you have the variable heavy, just the blue part, and then right from here all the way down to the transmembrane, that is your constant heavy. So that's your typical immunoglobulin domain. Whereas this top part that you are seeing, the top portion right here, that is the pocket to which the antigen binds. And also it do contain a carbohydrate. Due to that, it is a glycoprotein. So this is the overall structure of the immunoglobulin. So the FAB binds to the antigen specifically, as we say that this is the FAB fragments, marks the antigen for immunological attack and activates the defense mechanism that can destroy the antigen. For example, we have seen opsonization for enhanced phagocytosis. That the opsonization is that, think about that we already talked about as an immune response by the serum complement proteins. If a bacteria is coated by the antibodies, it will enhance the phagocytosis. We have seen that. On the other hand, the FC or the constant mediates the binding to the host tissues and receptors on various immune cells. And this is the first component of the complement system as well. That means that this constant FC region needed to be recognized by the first complement system in the classical pathway to trigger the immune response. So FC do have a function as well. So here we are looking at 2 of these immunoglobulin classes, and there are 5 classes of the immunoglobulin. You have a detailed list of those in your textbook. You can see that chart, although very much detail is not needed. So the first one is the IgG or immunoglobulin G, and 80% of your serum immunoglobulin is the type. This is the most common one. And this is required for opsonization, neutralization, activates complement. And only this is the Ig or immunoglobulin can cross the placenta for natural passive immunity to the neonate. And along with that, all the other though, which is secreted in the breast milk, can go into the newborn, but that is different. But only the IgG can cross the placenta, so that can protect the newborn as well. So again, this structure we have already seen, the variable light and the constant light chain. Then you have a variable heavy, and then all the way down to the constant heavy chain. Then you have a variable heavy, constant heavy, and the heavy chain, if you just want to remember, it's about 450 residues. So you can see that it is pretty large as such. On the other hand, your light chain is made up of about 212 residues of amino acids. IgD, on the other hand, is a part of the B cell receptor complex we have seen and signal the B cell to start the antibody production or convert it by this signal into a plasma cell. On the other hand, we are going to see that IgM, we talked about that, is a pentamer arranged in a pinwheel. This is the first Ig in all immune response, but instead of a pentamer, a monomeric or one of these IgM is also the part of the receptor complex of the B cell receptor. And then IgA is the secretory IgA or IgA. They are monomer, and two monomers form a dimer. So one IgA molecule will have two individual monomers to form a dimer secreted across the mucosal surface, and they are present in your tears, saliva, in the mucosa-associated lymphoid tissue, and helps in the immune exclusion as well. So just an overview of that, and we're going to come back. IgE, on the other hand, do have the lowest immunoglobulin in the serum level, and this is elevated in parasitic infection and allergic reaction. So what happens, the mast cell binds to the Fc portion activated to granulate vasoactive granules when Fc portion binds to the allergens. What do we mean by this? That suppose you have this IgE circulating in your serum. When it binds to an allergen, that is activated, and that particular IgE, due to the conformational change, due to the binding of the antigen, can activate the mast cell. When the mast cell will degranulate, means it will release the granules inside the cell, and you know that what these granules are. For example, these are your histamine or prostaglandins, vasoactive granules. That means this will cause inflammation, mainly by vasoactive means that they can dilate the blood vessels. That means vasodilation. That's why they are vasoactive. And this is done by the mast cells, will give you allergic reactions. So here is a structure of the pentameric IgM. Each one of them is an individual IgM, and this one monomer of IgM is present as a part of your B cell receptor. But in a pentameric IgM, you have 1, 2, 3, 4, 5. And why you have this in a pinwheel and why this is produced first? Think about that each one of them do have two antigen binding site. This is one antigen binding site. This is one antigen binding site. Because you all know that from the previously, FAB binds to the antigen specifically. So when we look at that, see that right here, FAB, this is your one antigen binding site. This is your another antigen binding site. So each of these immunoglobulin molecule can bind to 1 and 2 antigens. So if you look at this IgM, each one of them can bind 2. So you can bind 10 antigens at a time. So it makes sense because this is produced first so that you actually produce a large amount of the IgM. You surmount an initially very, very strong attack towards the pathogen. So in the first place, they cannot do or go farther. On the other hand, this is your secretory IgA in the dimer. You have one monomer of the IgA, another monomer. They are connected by a secretory component, and these are present in your mucous membranes, tears, saliva. Very, very important, and keep us completely protected to our daily ways through all the mucosal opening, bodily opening, as well as through our eyes. Then this is your IgD, which is the structure present in your B cell receptor, and this is your IgE. So when you are seeing that there are some specific dots, that what you are seeing, that those are your specific moieties, which are the glycoproteins that you are seeing. So these are heavily glycosylated as well. So the antibody synthesis and secretion can be evaluated as a function of time. So there is a primary antibody response, a relatively slow antibody response when an antigen is encountered for the first time. But when you have a secondary at-point response, it occurs as subsequent exposure to the same antigen, rapid, efficient, and this is responsible for preventing the illness because the adaptive immune response remember a pathogen, and this is the basis of the vaccination. So your primary antibody response takes several days or weeks of lag or latent period after the initial exposure to the antigen, as no specific antibody is detectable in the blood. And after the B cell differentiation into the plasma cell, antibody is secreted. Antibody titer is the measure of serum antibody concentration. So sometimes the doctors say that we need to check your antibody titer. That means suppose you have taken a vaccination, the doctor wants to double check that whether you really have that antibody in your blood or not. As you can see, that IgM appears as the first antibody followed by the IgG. So let's take a look at how the antibody is produced and its kinetics. So right here, you have the first exposure to the antigen. And first, this is your total antibody, but see that what is being produced first. First, the IgM, those pentameric antibody is produced first, and then you have the IgG produced next. Then it will decline. If you have a second antibody exposure, see that immediately, super rapidly, IgG is produced first, and then IgM to some extent. So this is the very important part, that the kinetics of the IgM and IgG completely switches. That IgG, which is the highest amount of antibody which is produced in your cell or present in your serum, is produced as a rapid response, and this IgG will stop the infection to go farther. So here you can easily see that this is a B cell which will proliferate, and they will secrete the IgMs first, and then they will switch from IgM producing cells to IgG producing cells. It is the same cell that will produce the IgG, and at this time they will be converted into the plasma cells, and the plasma cells produce the IgG in a very, very large amount. So what is the difference in between these two? See right here, the IgM is secreted, again, not a large amount. IgM is not secreted such a large amount, but at certain time right here, these B cells are going to convert it into cells which will be secreting IgG. And we are going to talk about that. This is known as class switching of antibodies. So the class from IgM, it switches to IgG, which is the main protective antibody, rather than the initial shock by this large antibody pinwheel structure. Once it switches the class, it will again proliferate fast and produce these plasma cells, and these are the large cells will produce large amount of IgG, and the amount rises. But see that what happens in the very first place, immediately upon exposure, you also produce these memory cells right here. Some of the B cells from your initial exposure will convert into the memory cells. On your second exposure, your memory cells will produce this IgM, immediately will switch into the IgG, and it will produce quickly these IgG in a very, very rapid amount. So that's the reason that your IgG goes up very fast and you have a faster immune reaction, which will not allow the disease to happen in most cases. So secondary exposure to the same antigen, B cells mount a heightened memory response characterized by a shorter lag phase, more rapid log phase, longer persistence, higher titer of antibody, and production of antibodies with a higher affinity of the antigen. Very specific type of antibodies. The next portion that we are going to see, although we are not going to go into the detail of the molecular biology of that, because that is a entirely different area that we can discuss all day long, that there are 4 mechanisms that contribute to generation of the antibody diversity. The rearrangement of antibody gene segments, which we call as a combinatorial joining or permutation mutation kind of joining, are split and interrupted into many gene segments. And generation of different codons during the antibody gene splicing, and the different codons are created in a process called the splice site variability. And we're going to see that. And also, these result in something in these cells which is called as a somatic hypermutation in these particular cells. So constantly, these will lead to diversity of antibodies, and you can easily check in your textbook. There is a small table which shows that how many different combination of these antibodies can be produced. And we are saying that the diversity of these can produce 10 to the power 13 different combination, trillions of types of these different types of antibodies. And it's kind of mind-boggling that what they can detect. So what is the combinatorial joining? So the segments of different region of these genes which make these antibodies are clustered separately on the same chromosomes. What they are? The exons that code for the constant region. So you have some constant region, exons that code for the variable region, and then joining the J region between the V and C region. So these are the exons, and in between these, there are all these different introns. So that's the very important part. All the exons are constant regions are joined, spliced together to one segment of the variable region. And here you have some specific enzyme which are called RAG1 and RAG2. These are the recombination enzymes which are responsible for joining one V gene with one J segment, and all other V and J regions are cut out of the DNA and are lost to the cell. So one specific cell in its chromosome is going to create many of these V and J region combination by splicing. And that will be a specific clone, and you have all these variable clones. And suppose by combining these, there are some B cell that's developed that is reacting to your self-antigen. That will be deleted. Only the combinations which will not react to your self-antigen will be selected in the bone marrow to come into your lymphoid tissue, which can recognize only foreign antigens. So let's take a look at this. So in the chromosome, you have this multiple V region exons. Each one of them has an intron in between them. Then you have these different J regions, and then you have a constant region. Now what happens that at this point, you have this combinatorial joining. So in here, this V2 is chosen, that is joined to this J3. Then you have the other regions coming in, and then you have the C region. And then again, by these splicing and translation, you can see that all the different areas are taken out, and you have only the V2, J3, and C. So think about that. You have so many different V regions, so many different J region. How many different combinations can happen into the variable region? Remember that the constant region is the same in all different classes of antibodies, because this doesn't bind to the antigen. This V and the J region is the variable region, which is responsible for binding the antibodies, and all these combinations are going to lead to this diversity. But remember that not only the amino acid sequence of these differ, but along with that, you also have different codons generation, and also the splice site variability and somatic mutation can lead to all these different combinatorial joining. This is for the light chain. On the other hand, so this is the constant region of the light chain. This is the variable region of the light chain. Now you have the heavy chain. Same way, the V and the J regions are joined to a third coding region called D, diversity sequence. And we're going to see that how the antibody class switch happens, and this class switching only happens of a difference in the heavy chain. So the V, then D and the J regions are joined with a new constant region that encodes a different class of antibody, IgG, IgA, M, C region is deleted along with other intervening sequences. So the difference is right here, see that for the light chain, the constant region is the same in all different classes, IgA, D, E, G. For the heavy chain, the constant region is different in different classes. That makes the different classes. So let's take a look. So for the heavy chain, you have the V regions, then you have the J regions, and then you have also an another region which we call as a diversity regions. So that gives you V, then D and the J combinatorial region. But instead of just one constant region in the light chain, here you have a C mu, you have a C delta, and you have C gamma. Depending upon what type of C is joining with the variable region, either mu or delta or gamma, that determines what kind of antibody it will be. So C gamma, a specific type, C delta specific type, C mu, that is called the class switching. So that is very important as such. And these are some of the important regions. So the splice site variability is the V and the J joining region can produce polypeptides with different amino acid sequences. We already talked about that, and this gives you the more diversity of that. And along with that, there are some region which gives you some of these hotspots which are susceptible to high rate of mutation. And this produces these antibodies with different epitope recognition. So now say for an example, what happens that initially it says the B cell DNA after first DNA splice will produce an IgM which contains this mu region. Then there's a particular enzyme that is activated when there is a first exposure to the antigen, which is known as activation-induced cytidine deaminase or AID. So this enzyme does some very interesting thing. Right here in this region, it converts the cytosine to uridine, uracil. Once you have an uridine residue or uracil residue, there is no uracil in the DNA. It's only the component of RNA. So that is going to cause a break in the DNA, and that area is going to be cut out. So the introduction of the U's near the C mu cuts this portion out. So what happens that with the V2, D2 and the J3 region, no more a C mu region is going to get joined. Instead, that is spliced out. And now you have a C gamma is coming in and it's joining with this. And this will lead to formation of this IgG. And now you have the class switching from the IgM to the IgG. Important thing, the same cell, which is the same B cell, initially was producing IgM. Once it is activated and cuts out that C mu region or the constant mu region, this is deleted from the chromosome, and that's why once it converts from this class switching from IgM to IgG, that particular B cell will never be able to produce IgM once again. So this cell will now become also, after the activation, memory cell. Next time it sees the antibody, it will not produce IgM. It will directly produce the IgG straight. And now it will make sense right here. You can easily see that why these cells, initially the new cells, will also respond by producing IgG very, very rapidly with a very short lag time. So this is the story of the class switching. And we already talked about how the antibody diversity can be increased over the period of time by the splice site variability and somatic hypermutation. So 4 of these things are important. So just remember that. What are these 4 mechanisms? That's your typical questions. And detail of that is not going to be necessary. And the clonal selection theory, remember that this is a repetition of the same thing. Body forms a large diverse B lymphocyte pool that can bind to a large range of antigenic epitopes. Cell-reactive cells are eliminated at an early stage of development. This is called the clonal deletion. Encounter with the antigen stimulate only those B cells that recognize and bind antigen, and stimulated B cell proliferate to form a clonal population. All have the same antigen specificity. So you can see that lymphoid stem cells can produce a lot of these different varieties. Some of them reacting with the self-antigen, they are eliminated clones. You have all these varieties. Now, suppose an antigen comes in which activates a particular clone. This particular clone will activate, and all these immune response will going to produce the same clone. So they are going to produce the same antibody so that it can bind to the same antigen or the epitope. Next time, if there's a different infection, it can lead to another activation which will recognize that specific antibody. So this is the lymphocyte clonal expansion. So how the antibodies doom the antigens? Bind antigen with a great specificity. Microbes and their products can bind to cancer cells. Invade foreign material can be bound by neutralizing, opsonization, immune complex formation. And here we are looking at what are the different types. So what are the consequences of antigen-antibody binding? So let's take a look at opsonization. We already know that if a bacteria is coated by these antibodies, it helps the macrophages or the phagocytic cells to engulf those faster. These are opsonized bacteria engulfed more readily. It will also lead to the activation of the complement pathway. That's also very important. That will lead to opsonization. The second thing is precipitation. Large antigens, they can bind to these epitope and form this lattice-like network, and that can precipitate these antigens. Usually, most of the time, these precipitated antigen-antibody complex can be taken up by the phagocytic cells for the destruction. Also, it can bind, say, for example, a virus or the neutralization. This can block by binding. This can happen with the different toxins as well, not necessarily a virus. On the other hand, complement fixation, remember that also that once these different antibodies bind to a pathogen, it can lead to this complement activation by different pathways, classical, alternative, or lectin binding pathways that can lead to this membrane attack complex to be inserted onto the membrane of the bacteria, which can lyse these cells. On the other hand, you can see that this is an agglutination, that if it's a large pathogen, here it is happening to a small toxin or antigen. This is a cell-free molecule. Remember that. These are much smaller. It can happen to this, which we call as a precipitation. Agglutination, on the other hand, when large, like a different bacterial colony, they are cross-linked to bacterial cell antigen. This basically happens in our eye all the time. So it's kind of a gross, but it is important that think about that you have this crust in your eyes. Sometimes you wake up in the morning, and many times this crust will have a combination of these agglutinated bacteria which got into your tears, and sometimes they will be lysed by lysozymes as well, lysed cells as well as agglutinated bacteria. These all make up those crust in your eyes in the morning as well. So toxin neutralization, inactivation of toxin resulting from interaction between specific antibodies, and these are all very self-explanatory as well. Viral neutralization, we already see that IgG, IgM, Ig antibodies can bind to some of the extracellular viruses, inactivate them, and also the fixation of complement component C3 pathway for the classical complement pathway helps in the neutralization process as well. And also, not only that, it will also trigger the inflammatory reaction. Very, very important for that. Opsonization, we already talked about that, detail, self-explanatory. And also the immune complex formation, and we already talked about the two things. One is the precipitation, which is the smaller molecules, whereas agglutination is a large immune complex formation, and this is readily phagocytosed by the free antigens. So this is very important as well. This is the basis of many immunological assays. Say, for an example, when we, again, not going into the detail of that, some of these blood test, ABO blood test is done, which is due to this agglutination reaction. But again, there the antigens are entirely different, and we're not going to talk about that here. So this is your immune complex formation. So one of the important thing that I just reading right here, that it is a tough material, but try to look for the connections between the innate and the adaptive immune response throughout the material to help you understand the whole. That's the main thing, and practice your practice quizzes and you should be all right. But this is a little bit tough material. There is a lot of information in there and a lot of reasoning and correlation. That will be the end of the last chapter of module 4, the chapter 33.